Hello everyone, I'm MEGHA VINOD, currently pursuing my BFSC degree from College of Fisheries, Dholi under Dr. Rajendra Prasad Central Agricultural University. Today, for all of our Being Fishco team members, I'm here to talk about something which is very novel as well as important too. As we all know, since humans have started exploiting the resources, they've always been greedy enough to fetch more and more from the same, and thus today finally we have landed at a situation where we have nothing left in our hand. We have absolutely nothing. Currently, most of our resources are either at the verge of extinction or are overexploited to such an extent that its replenishment is totally unimaginable. Therefore, it is high time that we ourselves take some important steps to replenish the resources so that future generations could be sustained. So, today, in this regard, I'm here to talk about a very novel method which has been discovered by scientists across the globe for the conservation efforts of especially the fish species that is a topic of our concern. And this particular method is currently being under use in the developing countries for its as a part of its conservation strategy and is getting shifted to the developed countries too as a means of management. So, let's not lag and let's directly go into the topic straight away. So, as the title suggests, out of the blue, it's time for a revival. It's conservation for a life. This is actually the cycle. Resources, human exploitation. Resources are there abundant. Humans are also there with their brain very sharp and strong, and thereby they're exploiting the resources to such an extent that today it's overexploited and almost extinguished. So, so here today we're going to talk about something which is very interesting actually. As we all know, every species have got its own genetic material and they leave their genetic material behind in the habitat that they live as something which is very interesting to know, isn't it? So today here we are going to talk about something called as eDNA, that is environmental DNA, using which we are going to manage the resources which are present in our current habitat and environment. Environmental DNA is basically all the DNA which is present in environmental samples. It can be both intra as well as extra organismal. As the ability to observe macroscopic organisms living in aquatic ecosystem is somewhat limited, researchers have recently harnessed the potential of environmental DNA for the detection. The eDNA allows us to detect the presence or recent presence of species without the direct observation or capturing the entire organism. Thus, it is something called as non-invasive method of monitoring fish species. Traditional fish monitoring system that we have been using is somewhat inadequate or provide less than ideal information that we need for conservation purposes. But eDNA is something which is very reliable. It has got very high probability of detecting species. Thus, in conjunction with the traditional surveys, this could be used or it can become a very powerful tool to enhance the detection rate and increase the confidence in monitoring the results too. This is also a less costly and non invasive method and more accurate, however. It is an important tool for aquatic ecosystem restoration planning to regular monitoring of the species composition with a focus on detecting organisms too. It can also give an idea about the paleolimnological history of the threatened species and thereby helps in reconstruction of such species into the habitat. It can be also used to determine the colonization history of species and structure of their ecosystems, aiding in the identification of native diversity and introduction of non-invasive or non-native species. Due to the high detecting rate and low density, eDNA based monitoring is especially useful for early detection and monitoring of invasive species and also helps to inform the fish management authorities about the species concerned and also enhance the likelihood of success of containment actions for invasive species. These are about the advantages or the benefits of this particular technology, and now into how we can pursue this technique for the benefit of the fish resources of the entire world. It includes processes like sample collection, eDNA capture, eDNA extraction, quantitative and qualitative analysis of the eDNA, statistical analysis and model, and comparing with the reference sequence database. So one by one, let's see into each and every step of this. So first step is all about sample collection. Sample collection, as we all know, we use different methods for sampling, and here we basically go for graph sampling and composite sampling. The volume collected also differs. Actually, it varies from 15 ml to 5 liters, or maybe it can go up to 68 liters. And the sample that is collected may be related to the abundance of fish DNA in the particular water sample. Generally, we go for 1 to 2 liters from the regular streams, rivers, lagoons, and also the sea water for collecting these samples of the particular DNA of the fish species concerned. Next is about the eDNA capture. The main methods used is filtration, precipitation, and centrifugation. Filtration is the most commonly used method where we go for membrane filters. On-site filtration is also possible using Stereox filter, which is somewhat handy and effective because it can be used in places where the laboratory access is not available. The filtration method has got advantages in dealing with larger bodies of water to achieve the higher amount of eDNA. And it can use the materials or the membranes with pore sizes from about 0.2 to micrometer to 3 micrometer, depending upon the usage or the need of the hour. Now it can be also made, the material is actually made of glass fiber, cellulose nitrate, or polyether sulfone. They are generally commonly used, and they have the chemical properties of absorbing the DNA and proteins onto their surfaces, thereby improving the efficiency of DNA collection. After filtering, the filter samples are either stored in frozen or the before extraction method, or placed in ethanol for up to 4 days without significantly affecting the DNA copy numbers. So it should be made very clear that the DNA that has been collected should be kept as such for preservation or for storage purposes for later use, because it may sometimes go on replicating if suitable medium is provided. So that may actually affect the studies that we are concerned with. So it should be kept as such in the maximum possible way. Now, the precipitation method is the next method of eDNA capture. The precipitation methods actually requires adding ethanol or isopropanol with sodium acetate to the water sample. But this method is only feasible for about small volume of water, which is less than about 30 milliliter. So next is about the method called as centrifugation. It can be applied. This method can be applied to larger volume of water, but it's not commonly used. Because it is difficult to apply for larger bodies of water, so it's not commonly used. And this method and combining this method is also another way of using another way of DNA capture. That is, different methods can be combined, like precipitation with ultrafiltration method or the precipitation with filtration method can be combined for better eDNA collection and capture. The next step of this eDNA sampling or the entire process is eDNA extraction. The most common commercial kit which is used for this extraction is DNA is a blood and tissue kit, or the power water DNA isolation kit. The choice of extraction kit, it can be depending upon the interpretation significance of the result that you require. So in addition to the kit method, liquid phase separation methods like using acetyl trimethyl ammonium bromide method and phenol chloroform isoamyl alcohol method can also be used. So I don't think we at this particular stage need to know about in detail of each and every step of this. I'll just give you an overview of how the entire process is done. And in last, we'll talk about its significance and conservation of the fish species. And that is actually the major concern of our today's talk. Next is about the qualitative and quantitative analysis. It can be actually eDNA analysis method can be used for target species analysis. It can be used for population analysis, and also it can be used for multi-species and community analysis. So basically, target species analysis is about amplifying and detecting shorter fragments of the eDNA which is released by a target species, and it can be done using PCR with species-specific primers. In species-specific testing, actually, the eDNA is primarily detected by the methods like gel electrophoresis, quantitative PCR, nested PCR, digital PCR. The presence or absence of fishes in the environment can be explored by these conventional methods, but generally we today go for real-time qPCR method. That is the most probably used method today. Now about population analysis, DNA can be used as a population genetic tool but actually it is suffering some sort of technical challenges which is not yet sorted out. It's under research because eDNA is released from different individuals within the population. They coexist in water sample. The eDNA analysis can be therefore extended to assess the intraspecific diversity, and detection of eDNA can be used as a reference for population attributes such as abundance, distribution, and biomass. But there is some inhibitions towards this particular analysis. So the scientists are actually sorting out the things, and we hope it will be somewhat fine and will be coming out in a better way in the recent future itself. Now, as next is about the multi-species and community analysis, the eDNA usually provides information multiple taxa too, and therefore particularly valuable for studying multiple species and community dynamics. Real-time multiplex PCR uses a set of species-specific primers and probes that are labeled with different fluorescent dyes for each target species, and thereby we can approximately 2 to 5 species can be detected simultaneously within a single reaction. Next is about the eDNA concentration models, that is a more statistical models which is related to fishes. eDNA is divided into three, that is eDNA concentration models, quantitative biomass estimation models, spatiotemporal distribution prediction models, and the abundance is and these are different models, and using these different models we can actually construct the DNA and its abundance in quantitative way. Next is about the reference sequence database and primers. It can be then exact identification is entirely dependent on the breadth of sequence DNA that we obtain from a particular water body. The obtained DNA sequence can be compared with some public reference database which is already available with us at the National Center for Biotechnology Information Database, that is NCBI, or the European Molecular Biology Laboratory Database, a Barcode of Life database, a Fish Barcode of Life Initiative online database, and the mitofish database. So these are different databases which is having all these things. So already preserved the sequences, so we're using it. We can compare with the DNA sequence sample that we have with ourselves, and thereby we can identify the fish species available in a particular water body. The DNA barcoding sequence from local fish fauna can also be used to build a local reference sequence database which can aid in future in more accurate taxonomic identification. So that can be done using Sanger sequencing of tissue samples. The molecular markers development is a critical step for fish DNA detection because the markers must be able to ensure accurate detection of specific targeted species or assemblages. So that is a very important step, and today mostly we use nuclear ribosomal RNA etc. for estimating the population diversity and quantifying the integration levels of invaded population. So the researchers have already designed universal fish primers. 12 is rDNA, site B16 is rDNA, and so. So we've already in hand. Now let's talk about the important factors. This is an important step actually. That is the important factors that is affecting the fish eDNA surveys for every method of conservation, or for any method of concern. It is actually it will also means influenced by different factors or conditions, and that condition will determine the efficacy of that particular method. So here we have both biotic as well as abiotic factors that is affecting the process. So let's see one by one. Understanding the factors actually influencing the detection probability could have significant implications. Okay, eDNA degrades while in transport either via biotic or biotic processes. It could be like sedimentation and biofilm formation, or interaction between the two or more of the other methods and all. To date, an integrated framework that identifies the factors which is controlling the fish eDNA movement and fate in lentic and lotic environment is actually lacking, but we are actually trying. The scientists across the globe is actually trying to figure out the problem, and they are trying to produce a final culminating procedure so that we can use this method in a very wider scale. The characteristics of DNA is something like molecular conformation length, its correlation with organelles, membranes, and all. The abiotic factors like UV radiation, dissolved oxygen, pH, the biotic factors related to the organism itself are the biological and environmental factors. All these are the influencing factors affecting the persistence of eDNA. Actually, in this particular method, we rely upon the persisting feature of the environmental DNA or the DNA of a particular fish species and all other indicator organisms, and thereby we survey the presence or absence of the near presence of a particular organism. Thus, we derive the conclusions from it. So, the factors that is influencing such persistence behavior of the DNA is very important, and this very important to know, and also very important to be concerned in this particular survey. So let's see the abiotic factor. After being shed into the environment, the eDNA will persist in water for days to months. The hydrological conditions like water temperature, UV radiation, pH, salinity, substrate turbidity, and water chemistry all influences the persistence. Hydrological processes can both influence the distribution of fish eDNA in lentic and lotic system. The fish eDNA exhibit patchy distribution lentic, and they lack sufficient mixing. Because they lack sufficient mixing, the distribution of fish eDNA can also be affected by hydrological conditions such as turbulence in the river and stream and strong currents of marine waters. Now, about the stream flow direction is also influential on the eDNA detection. Water temperature, it not only affects the release of eDNA but also affects the degradation. The temperature is a main driver of the eDNA distribution, with warmer regions having higher eDNA concentration. The higher temperature actually directly increase the DNA degradation by denaturing the DNA molecules and indirectly degrade eDNA by increasing the enzyme kinematics and microbial metabolism. The degradation of eDNA can be accelerated by disrupting the DNA base pair bonds by UV radiation. With the characteristics of high salinity and ionic content, high pH, and more stable temperature, marine systems have been shown to promote eDNA preservation more than freshwater systems. So there are different values for the decay rates in freshwater as well as the marine water systems. It differs. The sediment play an important role in eDNA preservation too, by both binding and protecting the DNA molecule from other foes and by binding and inactivating the extracellular nucleases. So it is a positively affecting factor. Next is about the different abiotic factors. The abiotic factors in the sense the number of organism, life stage, behavior, body size, metabolism, stress organisms are under, and seasonal migrations will be affecting the strength of eDNA signals. The fish biomass juveniles generally have higher rates of eDNA release than the adult fish because the behavioral metabolic changes can affect the eDNA production too. The fish eDNA production increase significantly during the breeding season. So during spawning period, multiple sources of genetic material is released like blood, urine, fecal material, epithelial cells, and gametes. So there will be an abundance of species, could be overestimated. Okay, and this could be overestimated during that period. So hence it should be made sure that we don't go for sampling, especially during the breeding season, because there is a chance of overestimation and there will be a irregularity in the results obtained. Now, the body surface area is also considered to be an influencing factor because a small fish stock releases more genetic material than a single large of the same quality. Fishes with higher metabolic capacity have got high cell production and division rate, and thus can lead to higher DNA shedding rate. Human activity and predation pressure can also alter the stress of fishes, which in turn can affect the release rate of eDNA. The eDNA concentration is intermittent stream is influenced generally by the seasonal variations too, also by the migration of fish species. Moreover, the allogenous DNA might influence the result of eDNA monitoring too. For instance, like the piscivorous birds, they are one of the potentially important source of allogenous fishes in the aquatic ecosystem. So that is also a major concern. Now about the biological environmental factors like the microorganism and the extracellular enzyme activity is another important factor affecting the degradation and accumulation both. The presence of biofilms can also significantly increase the initial decay of eDNA. Although the eDNA method has got many potential applications, survey will actually be need to carefully design the given number of potentially confounding factors. The knowledge of the relationship between the eDNA production, its persistence, and influencing factors can guide an effective sampling strategy and interpretation of the results, and this is very important matter of concern. Now about the different applications of fish eDNA, it is achievements are different. It is used in species monitoring, it can be used in population monitoring, it can be used in multi-species and community monitoring. This was mentioned before. Like it has got different applications. A basic DNA, a fish eDNA analysis has got conservation objective, and it has got the surveying objective, it has got the restoration objective, it has got the analysis objective, that is this continuous species monitoring, about the population monitoring, multi-species and community monitoring. So all these are the different features that we actually require in this current scenario of continuous devastation of the aquatic habitat and its resources. So now towards the ecosystem and the environmental conclusions, like eDNA can be used in both active as well as passive surveillance of monitoring species in the ecosystem. It requires considering the correlation. Actually, it requires such a consideration of the correlations between the fishes and predators and the competitors and the prey. So, different fish-based biotic indices like the IBI can be derived from eDNA data to reflect the integrated aquatic health condition also. Thereby, it can be used as an effective assessment tool for managers to formulate conservation restoration strategy. It can also be gradually applied to solve the hypothesis-driven scientific questions like regarding relationship between the biodiversity of fish and ecosystem function in relation to external anthropogenic or natural forcings. Now, eDNA monitoring can also have the potential for evaluating reintroduction of rare fish species which attained a good ecological status of fish freshwater bodies is given by biological indices and all. Thus, fish eDNA approaches have got a very potential for ecosystem health management too. Chemical substances which cause fish mortality can also be reflected by change of eDNA concentration. In recent years, actually, some studies have used fish eDNA to detect the mortality of fishes after chemical treatment. Fish eDNA trends could be useful thereby as an indicator of carbon dioxide pesticide efficacy, which is currently being developed for non-selective fish population control. So there are also different up-to-date study tools for fish eDNA ecological monitoring, like about the eDNA monitoring devices and CRISPR nucleic acid detection, meta-omics technology, and fish eDNA studies. They are the recent advancements in this. So, towards a concluding note, we would like to say that the eDNA technology will be one of the major tools used to describe, model, and predict fish biodiversity in space and time. The monitoring of fishes by eDNA can actually accurately identify species while avoiding the inherent bias in traditional biological monitoring. Such methods are actually changing the basic research way of collecting ecological data and monitoring the ecosystem. The development of standard operating procedures, including sampling and everything, will improve the accuracy of this technology actually. Full clarification of the influencing factors across different environmental conditions can also help to propose a specific solution instead of employing a one-size-fits-all strategy for eDNA study. The modeling of uncertainties associated with eDNA concentration for quantifying the abundance is still in the developmental phase. The up-to-date tools, especially in situ autonomous monitoring device, are recommended for the fish eDNA research, as these will pave the way for generating live monitoring of fish genetic information. The application of CRISPR nucleic acid detection meta-omics technology may also provide more effective and broad note on fish diversity. So currently, only the developing countries are either facing, as we all know, not only the developing countries, even the developed countries are also facing severe habitat degradation, and thereby they forced to, or actually they are not forced. It's only because of their own work that they are being forced to go for ecological environmental protection initiatives and all. So, as a possible paradigm for fish monitoring and future method of evaluating the health, And this eDNA technology has to be promoted worldwide because it is something which is very cost effective. It is something which is non-invasive. It has got many different applications, like it can be used in species level monitoring, the population level monitoring, its alterations at the population level, the multi-species interactions. It can also be used for identifying the monitoring the integrating organisms, and it can be used for health of the aquatic ecosystem analysis and its management. It can be used for the invasive species, its control and its effective management. It can be used for habitat restoration, also for the restoration of different extinct species, and many are its advantages. And being a very non-invasive and very cost-effective method compared to the traditional method, this can definitely be a future scope in this particular conservation strategies adopted worldwide. So, once sufficient experience and data have been accumulated, this method can become a routine in fish conservation research and will provide, therefore, a scientific basis for its formulation of the ecosystem protection policies, too. So, thereby, let's go for something which is scientifically proven and innovative, and only such methods can help us in the future for the protection of the resources that we ourselves have depleted. It is the entire thing behind it is a human brain. It's a human brain which have destroyed, and it's a human brain which is developing methods to revive it too. So if we take the responsibility, if we remain concerned about the resources, its utilization, exploitation, therefore we can prevent such things from happening. That is, we can actually prevent the need for such conservation efforts if we are actually becoming more responsible. So that is a need of the hour, and hope you guys have enjoyed the session, and thank you for your patient listening. That's all about today's talk. Thank you.